Ok. E niente, questo è il possibile dagli spiriti che si stacca e si riattacca da sotto. Ok, a posto. Ciao. So he has a bachelor degree and master degree in biology and so on, so on. He has a master in CBRM protection just for you, me, and next year Dr. Rao, sure. And he also had a PhD in industrial engineering. He works as a senior chemical researcher at Thermo Fisher in Ferentino. And I think that you will enjoy his lecture this morning. And please be very focused on his lecture because some questions or maybe can come from his lecture. So enjoy it.
You can remove me.
Please. So Marco, the floor is yours.
Thank you. Ciao Alba. So, good morning, everybody. So just say, Alba, I am Gianmarco Ludovici. I am a member of the didactical board of the first and second level master in cyber events. I have achieved the second level in 2016. Maybe this day of yes, 9 years ago. So I am a biologist. My works in the past during the university was regarding viruses and microbiology. Sorry. Normally I have a low volume of my voice. I have to high my volume. Okay. No, I see. Okay. Perfect. And now my focus in this research was the use and prevention of the biological weapon and using a CBRN. During the week, I am a chemistry analyst and research at the Thermo Fisher Scientific. Is a pharmaceutical factory, and for 3 years I worked for a Moderna project because in my factory was product the vaccine against COVID-19, and now the project is over because the pandemic was classified as finished. So first to start, I want to know your background. Presentation, very few seconds regarding your degree or your name. Like this. Here. Sogene.
Master's.
Okay. With your professor thesis? Gismondi. And field?
Plant science. Plant science.
Ah, plant. I remember was the Camoni,
Yeah.
Professor Camoni, Lorenzo, my friend. Ah, Lorenzo. Okay. Grazie. Okay.
So my name is Ignacio Velardi. I am a physicist and I work in ENEA at the
Frascacci.
I'm a Ludovici. I'm a research physicist at ENEA.
I'm Laura Zampetti. I'm a lawyer and a volunteer of the Italian Red Cross.
I am part of the. I am a member of the Order of Malta. And CISOM. Yes, there is the. I know the Red Cross. Yes. All the same department, ok. Partiamo da dietro a tornare avanti.
Jan Lappela, former British Army captain, bachelor's in British management, and also paramedic, knowledge work as third as a final paramedic.
Congratulations.
Good morning, I'm Franci, I'm a chemist, and I'm a lieutenant of the Italian.
Oh, two colleagues, it's strange. Okay. And you study Roma 3.
Morning, I'm Ludovici, I'm a chemist. I'm a private consultancy company.
Ok.
Chemist,
Morning, I'm finished school, and I am civil engineer in the National Department of Fossil Therapy.
Good morning, Ilaria Lezzi. I'm an international security analyst working for a private company in Italy.
Okay, so thank you for your presentations. Now, the lecture of today is divided in two parts. I hope to don't bore you with the not complicated arguments. And the first, the most volume regarding virus and the use of virus like biological weapons. We will analyze different viruses and also the use of the biological agents in biological warfare. We will analyze the most recent pandemic with the SARS-CoV-2 and also analyze the epidemics of last year, for example, Ebola in West Africa, 2014, 2016, and other use of biological agent in the history, starting from the use of plug deaths that put inside into the city to spread the Yersinia pestis bacteria, and also the use of Bacillus anthracis, for example, in the USA mailbox after the September of 2001. Okay. So, during my lecture, maybe it's better not make a classic professor, but an interaction with students. And I ask you different questions regarding your knowledge of this argument. For example, not biologists, but do you know, for example, what is a virus? And for example, another difference between a virus or a bacteria.
The box where the RNA inside.
Also RNA? Always RNA.
DNA or RNA. It's only a box, it's not a life material. It's only a box. and bacteria is something that have a more complicated process.
Yes. Like your colleagues, they write. So the normally a virus is formed by a capsid, is a protein that contain. Good morning. That contain genetic material. So, for example, it could be RNA positive or negative, DNA, RNA fragmented. For example, influenza viruses. Influenza viruses have 7 pieces of RNA. Every piece codify a protein. Generally, there is a other layer that covers this capsid, and it's called envelope. Envelope is a starting come from of the infected cells, and the spikes protein that make the interaction between virus and host cells. But the principal difference, let's say your colleague, is that the dogma of life. Organism, it's an organism. If born, grow up, reproduce itself, and die. The viruses in general don't do this. Because if there are no cells to infect, virus die. It's for its reproduction have to use the cellular mechanism to reproduce itself. Okay. So this is the stay before the virus structure. So the nucleic acid inside, the protein codes, the envelope, and spike protein. So the virus particle, or called virion, is nucleic acid, protein, and lipid membrane is called envelope. Normally, not all viruses have envelope, and it's called naked virus. Also have capsid. Another type of viruses, I don't know if you know, is the viruses of bacteria, and it's called bacteriophages. So it's the first, one of the first viruses that identified and most studied to study the viral mechanism to infect cells, and also they destroy bacteria like a normal virus destroy our cells during the infection. So have a different structure. So bacteriophages viruses infect only bacteria, and have a protein like a tail that attach the surface of the membrane of the bacteria, like a syringe pull inside the nucleic acid inside the bacteria. The nucleic acid interact with the mechanism of the replication inside of the bacteria and replicate itself all the structure and destroy the bacterial cells and the other particle, the other bacteriophages can infect other bacteria. The same thing that make a plant or animal viruses. Another question for you. What's the difference between structure regarding a virus or a cells? A human cells, for example, not biologists. For example, the structure.
Much more low tension.
Yes, but there are few years ago in Austria have discovered the Austrian scientists discovered viruses. It's called mega blah blah blah, and it's like to the dimension. It's like a bacterial cells, like a protozoa. Yeah, it's the biggest virus discovered in nature. So this is the first. Also,
that can be passed through materials on viruses. No.
yes, but there's all of the organization inside the membrane. But envelope viruses have a part of cell membrane derivative from the previous infection. But yes, your interaction with glycoprotein or the system to ion channels, yes, viruses doesn't have this. Other?
Part of the cell out of the nucleus.
Yes, the organelles. Yeah, mitochondria, or for the plant chloroplast, air, Golgi. Yes, yes. And for these things, the viruses needs this part of the cell to reproduce itself. During infection, for example, you know how the protein are made inside the whole body, no? We have DNA. DNA is form mRNA, messenger RNA, and mRNA inside the cytoplasm can reproduce the protein in base of the genetic code. Okay? When viruses infect the cells, mainly things. So the genetic material of viruses are transformed into a messenger that can recognize by how ribosome. The ribosome create the different protein of the viruses. And good morning. And normally inside the viruses, inside the host cells, all the reproduction apparatus are blocked only for the viruses, and it's called virus factory to reproduce a large amount of the viral particle that can spread out the cells by budding or lysis, and other viruses, new particle product can infect other cell in the apparatus, right?
But I understand for generate new protein from mRNA becoming from virus.
Mm.
But the replication of genetic material of virus, where happens? Because, for example, in the cell happens in the nucleus.
Right. Good question. So normally, every virus generally have two different types of proteins. Have a structural protein that is necessary to create capsid, and also have the non-structured protein. This is, for example, replicase or other type. When its product use the genetic material of viruses to replicate, but always inside the cytoplasm of the cells,
Okay.
and create this apparatus that called before virus factory. And so
So
after
there are two lines of
yes, it's the same. Two line of production. One thing to production the nucleic acid, another to create the structure to protect the nucleic acid. They are assembling, and when go outside from the cells, take the lipid membrane and create this envelope, and can infect other host cells.
Yeah.
Is a two line that after
So it's self-assembled inside the.
Yes, but also in the same thing. It's called is a part of cells called virus factor. So viruses are specific to the host and also the host and also the cells of the host. For example, hepatitis viruses can infect the lung because the lung cells don't have the receptor of the liver that is the host of the hepatitis viruses. Also, it's normally I use this as example. For example, we have the flu viruses of dogs or cats or other animals, but we have not dogs or cats. We are mammalian, okay. But if I interact with this type of viruses, I can make the infection doesn't start because my cells don't have the same receptor of the other animals. This is important. More people say, but my animal have a cold and I can infect them. No, the same family. Okay, so they can only attach the specific cells at the receptor site using the glycoprotein projection. For example, rabies can pass it from animal to human. Do you know how this called this passage? Right, zoonosis. And the most pandemic in the nature during the human history is zoonosis. Spanish flu, SARS, Ebola are zoonoses. When virus of the type of species make a jump and infect other species. So I just say regarding this, but this image is beautiful. And this is a simplification of the virus replication. So first we have the virus, and it's called virion, infect the host cells and go inside. This is a normal animal cell with the cytoplasm nucleus. The virus is go inside by the mechanism called endocytosis. Go inside. The membrane and capsid of the virus are destroyed, and the nucleic acids are free inside the cytoplasm. Virus use the mechanism of the cell replication to replicate itself, generate new variant, and go out by budding, destroying the cells. So different viruses, this mechanism create disease. It's strange. Actually, we don't have an answer to this question. So some scientists think that the origin of the evolution are linked with viruses because a lot of the junk DNA that are inside of humans are derived from the viruses. How? because there are a lot of viruses, a type of viruses, for example, HIV, right, that can take its genome and with a protein that is inside your DNA. So during the million of years, a lot of these viruses infected different species and give this part of genome. But the origin is like the origin of the life. So we have the primordial growth, and maybe viruses, if you think, is a simplified cells, if you can imagine, and it's like a parasite. And but the origin, how it started, or the first viruses, for example, we don't know why.
About the relation, the kind of steps. So
Yeah, maybe of this is during but the first I can imagine is the bacteriophages, because the first cells are bacteria, and the nature create a way to destroy a lot of this.
But it's a sort of evolution.
Yes, it's an evolution to maybe it's a part evolution, but the also this evolution is linked nowadays, and some scientists, for example, think that a lot of the type of autism is linked to this an ancient DNA incorporated by viruses. I have a friend, a biologist friend, that have studied this kind.
Sorry. Time I studied biology was 17. And the virus enter in the cells and destroy itself to generate
Oh, it's open like a box, huh? And releases the genetic material.
but one virus. How many other viruses generates with this mechanism?
Depending from the viruses, but thousands virion particles. So, for example, there are viruses that. So, if you can imagine a cells like a little city, okay? A virion is like a car, and you can imagine the proportion, and some viruses can destroy the cells because it's filled, and the cell explodes and release a lot of virion particles, thousand. I don't have here, but there is a beautiful picture of a bacteria filled of bacteriophages few seconds before explode. So we have talking about this. There are 5 points to know. Penetration. So DNA, the virus goes inside the cell. Replication. I don't know. Replication. There are really the replication of the genetic material. After there is the synthesis of viral protein. New particles are assembled and released from the host cells. As you can read, a virus is considered to have a quick replication when symptoms emerge within the first 2 weeks. But normally we know that we are making in touch, in contact with people that are infected. Normally the infection can start in a few or 3 days. For example, cold. If I stay here, the first symptoms can spread after 2 3 weeks. Also depending of the immune system that can stop a lot of these or not. So this slide talk about the role of viral disease and prevention. A pathogen is a disease causing agent such as bacteria, viruses, fungi, or parasites. The prevention, because the viruses have a particularity. Do you know the difference between virus and bacteria, and also to the fact the bacteria we have antibiotics. So the word explained anti-bios, against life. In this case, virus is not a live, and the only drugs that we can use are the antiviral. But respect to the viruses, the antiviral are very low number. So one of the most known antiviral is aciclovir. Do you know? It's for herpes simplex, herpes zoster. Antiviral, the aciclovir is famous because block the replication of viruses at the level of the reproduction of the genetic material. But if we use every time aciclovir, have discovered different types of herpes that are immune to aciclovir. Like bacteria is after several years are immune to a large spectrum of the antibiotics. So a little parenthesis. I work in a pharmaceutical company, and in my company we product two antibiotics against MDR. Do you know what I mean? MDR, bacterial MDR, is acronym of multi-drug resistant, because today the efficiency of the most antibiotics is very low, because during the last 10 years, every time, I don't know, maybe in Italy, doctor, I have just a little bit fever, take a cycle of antibiotics, cefixoral, cephalosporin, and during the year, these antibiotics doesn't work. And we have to create a new molecule to against these strong bacteria. For example, my bachelor thesis works is regarding the Acinetobacter baumannii. Do you know? The only antibiotic that can destroy these this bacteria is colistina. And sometimes also doesn't work. The only is clear the surface and hope that your immune system can block this bacteria. Is a problem for the public health. And another a weapon for us to defeat viruses is RSV vaccines. Do you know, after we're talking about the vaccine, but do you know how vaccines works? Okay, but not in the specific. Normally, generally, how? Yes, I give you a attenuated viruses or a part of the virus, your immune system creates antibodies against these, and when, maybe not, but in the future, we make in contact with these viruses, I have the soldier that can defeat the, just know the the pathogen and can interact with him, and if don't protect you 100%, maybe can make you more safety respect to people that the first time that interact with the pathogen. So this is the, sorry if I talk about this, but because I worked for COVID vaccine, more actually a lot of people, I don't know why, but thinks that this vaccine also is another vaccine. No. But don't think that worked. But I can I worked with the first stage of the vaccine, so at the PCR level, some people, a lot of people that make the vaccine and also take viruses doesn't show the symptoms. And okay, I take the vaccine, I have take the immune from viruses. Not. It's false for every virus. But the problem, not the problem, the thing that fortunately some save their life for this reason. And another question regarding vaccine is normally to develop a drug, a pharmaceutical company works 10 or 15 years. Do you know this? When I discover a molecule, a drug, and I can take the pills inside the pharmacy, more or less 15 years. So the question was how this vaccine we product in 1 year and a half, 2 years. Do you know this? Do you know the answer of the question?
It was part of just started.
More or less, because first the technology about this vaccine, it's a old technology, starting from the early 19, 30 years ago. First. Second, the application is perfect for these viruses because fortunately SARS-CoV-2 is a positive mRNA, like the mRNA technology. And finally, the million of volunteers that jump the clinical trials. So normally when I discover a drugs, I have to experiment to mouse, rats, dogs, and monkeys. After a little group of people, large, the people that have the disease, placebo, not placebo, double spectrum, double blind, etc. But if I have a large amount of data, because more people, especially in England or in the USA, give the volunteer to test these viruses, I have a lot of data to can jump all the regulation of FDA, IFA, and the European Medical Association, etc. And the drugs, in this case vaccine, was used for humans. only a little regarding drugs. Okay, yes, this is no RNA, only RNA, non DNA. RNA have a polarity starting from, it's called 5 to 3. It depends of the right, the position of different atoms inside of. You can imagine the mRNA like una collana di perle, ok? Normally, in nature, the direction is 5 to 3. It's called in this way. And this it's called positive polarity. If you have the opposition from 3 to 5, it's called negative. The viruses that have negative RNA first have to transform itself inside the cell in positive polarity, because how our ribosome knows only the positive. Okay.
Negative to change in
Positive, and after is transformed. But also another thing. Thank you for the question. Normally, and this is fundamental. Sorry. Normally, not normally, for human or the superior animals, every single mRNA is codified into a one protein. One mRNA, one protein. Stop. The nature works in this way. Our nature. For viruses, because viruses have to, we have not a lot of space like normal cells. With regarding the virus with mRNA, one mRNA have inside different proteins. So a piece, one protein, another piece, another protein, etc. And all have information for all the proteins that can the virus use for replication inside. What time is it, please? Make a short breaks to take a coffee, yeah?
Yes.
Prendiamo un caffè?
Lavorando dal fine della mano. Siamo ancora. È un po' come una gli aspetti del lavoro che piacciono di più, che piacciono di meno. Però sono una cosa che noto. Noi siamo quelli di visione, poi diverso. In un ente di ricerca le scadenze dovrebbero essere dettate dai progetti. La possono non può andare poi stanca, non può andare poi stanca perché questo è le cose. Però collegare, però diciamo che ha fatto lavorare la cosa e tu vuoi dormire, magari ti entra fuori dalla scadenza, dai, chiaro. Ma sono così che dove mettere in base alla reazione. Prima noi non carico quando ho visto questo che vedrò domani, il quarto dice perfetto, per dirti come siamo visti. L'ultima, tutto sommato, ha dato ha velocizzato leggermente le cose perché effettivamente ho trovato tutta una serie di emergenza di caraccia del 23 perché comunque erano pronti, comunque il circuito si era voleva provare, quindi ci poteva essere. È scritto. Sì, avevamo parlato. E poi la legge politica il piano di approvato un piano. Per fare un piano ci dico 25 anni per farne uno e poi ragionando se non faccio neanche il tempo ci spiego per farlo 3 anni ma poi quando lo faccio nel frattempo si crea il filmato di quella delle leggi di dare le scadenze ma non le spiegare ma perché? Non è soltanto una gara. È un po' poi arriva il voto per oggi perché lo pandemico la prima 2006 ma ci sono 3 anni poi da lì poi però poi nessuno non ho 20 anni ma l'hai fatto 15 anni è proprio velocizzato quei piani di emergenza che mi si spiega un po' poi non c'è tanto magari anche più troppo abbiamo dei diritti da fare non c'è da fare può fare cambiare perché veramente non va a legge scenamento posso dire affascinamento di routine. Quindi dice domani sono le facciamo. troppe troppe persone. Hanno il modo di partecipare nella comunicazione. No, il nome non me lo ricordo, ma dovrebbe essere contrario. Però l'ho conosciuto mese fa, sono andati alla giornata lì, non ci vuole. Era uno giovane, no? Quando ti presenti, ciao, c'è un vicepresidente, lo guardi, ti ricordi, ti ricordi, ma io non ne ho 40 anni, ne ho 45, io guardo, poi ti ricordo. Le scelte che fai sono tutte, sono... Sì, sì, sì. Sono... Le scelte che fai sono punto del cazzo. Queste sono delle... Poi siamo stati a... ci siamo io, per continuare a militare. Siamo lavorati in un periodo. E quando vado all'università invece posso stabilire delle università l'università prima era di ricercare e ho vinto. Dovevi prima essere ricercatore, poi come sono i ricercatori che sono un posto, aspettavano di 30, 40, 50, in realtà molto grandi, capire che un po' di due anni, si prende, e poi hanno cambiato la struttura che i ricercatori hanno legalizzato, però se passi 3 anni di identità, scusate, devo quando c'è il posto delle più non si quindi le persone adesso con tutta l'imbarcata che va in pensione ci sono più posti ah oh. Università per candidati che sono alle persone, se vai a Sapienza, lì, magari un po' di più di candidati. Non sono andato adesso, quando all'epoca c'è stata di giornata, subito dopo il 68, all'epoca era venuto del 65. Queste cose la vita, devo dire quelle mie scelte. E quindi dicevo, dai, il nostro lavoro, viene libero. Politica, cazzo. Certo, perché poi guardi le ufficiali, guardi poi a un certo punto ti posto, poi cercare una fare magari quando una guarda voi, una guardata e guardare il signore, io la guardo di là, poi regalo una portata da guardi, Quindi non cosa chiude tipo quella polizia scientifica invece era la cosa nazionale, poi dici, più di quel oppure c'è un qualcuno, c'è un paese, aspettando un po' di emigrare. Quindi, ero ancora giovane, poi sono che sono di età, a volte cambiano. Sono troppo giovane, sono un po' troppo anzi, che molto ritenere uno dei fortunati che era la squadra del giovane. State tutti i problemi, se mi dicono che posso no. E che fai la niente credo che chi viene dalla terra che fa lavorare la tua prende un po' tanto legalmente forse si stai nella propria terra magari sei il diritto come ha fatto quindi quando sei no, non hai dove temporale per lì, a bar, ma non è proprio così, non è proprio così, dovreste uno che comunque ha una certa età, quindi poi il dottorato, hai fatto che sei in infezione, posto di lavoro, che poi marcia. Quando certificati sono quasi gli anni, non c'è nulla di qualcuno, è nulla. Tanto certificati sono gli anni. Poi una partecipazione in caso, che la mamma sì, aspetta un po' di lavoro, arrivano della medica, ma non sapevo di avere un figlio, non guarda, la certificata, è l'appartamento forse non si ripete. Veramente, fuori e del prodotto non c'è un box, dice poco dentro, poco dentro, e fai solo dentro. Non devi chance. La bellissima fuori. Non hai visto se erano ieri era abbiamo oltre la fatta un po' non so che lui va che le sessi no, l'idea c'è
principale funzione. L'ho vinto la per rispondere alle domande di tutti gli altri, e mi dice ma che pensa che oh, guarda, devo fare una cosa, di
This is my place. This is my place. Facciamo il fondo del campione. ogni istante. Domani 20,
Vedrete invece. Enrico, tu ti chiedi perché diciamo, ma mi preparo, poi c'è tutto e non serve. E senti che le persone di
So, we can start at 11 o'clock. We make another break. And the second part, we will talk about mushrooms or fungi. So, I will go so too fast, and so I hope to not bore you. So, do you know this symbol, the biohazard? Okay. Regarding the dangers of different type of viruses, the viruses with in conformity of the CDC, the viruses or other biological agent is classified in biosafety level 1, 2, 3, and 4. Biosafety all and in base of the dangers of the viruses. Also, and this is change the structure of the laboratory. For example, BLS1 is a common university lab. And we use normal bacteria, for example, Escherichia coli or non-human virus. Biosafety level 2, we have a different door that have contained two departments. For example, a door in glass that divide with airflow that separate external environment to internal environment. And for example, BLS2, we can work with the flu viruses. BLS3, the most important is HIV, and you can cover with double gloves and glasses, 2 clothes, and 3 different levels of separation of the environmental. And the most important, that is famous, that you see in different movies, is BLS4. For example, in Italy, we have one lab of BLS4. Do you know where? Good. Lazzaro Spallanzani, the hospital Spallanzani. And maybe you remember when was the Ebola outbreak in 2014. There, Italian doctor from Guinea is transported by the aeronautic in the glove box, and there was say in Lazzaro Spallanzani, and have the compartment that can study this type of viruses. Another famous virus that is classified as BLS4 is smallpox, variola major, vaiolo. Okay. So this is the classification to CDC, and maybe this could be a question of your exam. So the identification is called category A, B, C, and I don't wanna read, but depends of the categories have different way to track treatment of viruses or bacteria. For example, Yersinia pestis or plague or Bacillus anthracis is category A. So this is an example of the category A according to CDC, and the most important is hemorrhagic fever. Ebola, Marburg, Lassa virus are the hemorrhagic fever that classify as category A of CDC. Also, smallpox, Bacillus anthracis, Clostridium botulinum. Do you know the botulinum is bacteria that create the most toxin, the most important and lethal toxin in nature. And also the category B, for example, there is ricin or a multi-drug resistant Escherichia coli called O147H7 and caused a outbreak in Germany from the salad. Do you know some years ago is discovered this variant of Escherichia coli, which normally is inside our gut. So one of the most important and famous viruses that classify as biological that can be used as biological weapon is pox viruses. So I don't know if you know your level of knowledge regarding the use of the biological agent to use as biological weapon, but for your knowledge, why the smallpox could be the perfect biological weapon? It's a simple question, very simple question.
Because for us, it's not dangerous. For other ethnies, it would be very.
No, because during 70, the smallpox was eradicated. Is the only disease, the only virus that it's called eradicated. Because for WHO, a disease, a virus, a bacteria, everything is called eradicated when for 2, 3, 4 years don't appear a case. Normally, we don't have smallpox in nature. Is right these phrases? Monkeypox, but we are not monkey. Someone yes, but we are not monkey. There are a lot of monkey of pox, but to different species of animals. For example, the cowpox, and we talking about this, is the way that discovered vaccine. But you can infected by a cowpox. You are not a cow, okay? The problem is that the variola major or major in Latin is there are not in nature, but in 2 laboratory in CDC of Atlanta and in a Russia laboratory, sorry for my pronunciation, but I don't know how to say in our language. There are 2 labs that have and study smallpox. Actually, there are people, for example, my parents, that have scribes here, that is the vaccination. But in 10 years, 20 years, a lot of these people will die. And actually, their immunization is low respect when making the vaccine. And if I don't have the viruses, I don't have the vaccine. And if I spread actually the smallpox, the death rate is 60%. So in this room, more than half will die. and we don't have a lot of vaccine to immunize the world. If we don't have a drug or a vaccine to contrast this, if a terrorist group can take a small amount of viruses, can probably kill half the population in the world. And this is deliberate use. After we talk about natural outbreaks, and this is more dangerous. So after we give you the slide and give you the information regarding these viruses, but the important was that how, thanks to smallpox, vaccines are discovered. Do you know the history regarding this process? This is know the history is important to know how the fit. So the doctor of the Danish king called Janner, Dr. Janner, see that the farmer don't take the smallpox. How? When take milk from the cow, a lot of cow it's infected by cowpox, and they touch the scrub of the cow, take the cowpox, but doesn't infect it. But the immune system create antibodies to cowpox that are similar, not the same, but similar to human smallpox. Okay? And it invented that take a piece of the rash of the cow and put inside of human. And this way, immunization humans. And starting from this, it's starting the era of vaccine. And it's called vaccine from Italian vacca, is called, and the name starting from this. In the 18th century.
Sorry, if we have the vaccine, why is a problem?
Vaccine regarding smallpox.
Yes.
We don't have vaccine. Some people that are alive have vaccine.
We have a solution.
No, because I say that to create a vaccine, I need a large amount of virus, because the smallpox vaccine is an attenuated vaccine. So to create vaccine, I need the virus. And if I have only two labs in the world, we can create a small amount of the smallpox vaccine. And this way is the problem. The same for Ebola. Ebola don't have vaccine or drugs that can contrast. And the other thing is fortunately regarding Ebola, the problem is that Ebola kill you very fast. But fortunately can spread with airborne, with droplets like flu, for example. Also with body fluids. So this is a weapon for us. Smallpox, fortunately, have a high mortality rate and can spread with airborne system. So the problem is this: if I don't have the weapon to contrast this, it's the way that could be a candidate to be the perfect biological weapon. If, for example, a terrorist group that can take this. So the famous Ebola, the filoviride, because the form is like, it's similar to philo, okay? is the agent that create Ebola virus disease. Ebola is a river in the center of Africa, and there was discovered for the first time in 1976. In Democratic Republic of Congo, ex-Zaire, if I can say the same thing, there are 5 types of Ebola virus. And the problem of this virus is that first, we don't have a cure to defeat. And sometimes in different outbreak of Ebola viruses, the scientists discovered that have a high mortality rate. Sometimes near to 90%. 90% of their death rates. And we don't have a solution to defeat this virus. The way of spread is through body fluids. So, for example, blood. And the problem is hemorrhagic fever. I don't know if you see how people die by Ebola. So your epidermis release blood. You can. I don't show the picture regarding this because it's not beautiful to see, but it's not a good thing. And the other strong problem is the cultural problem of the population where start the outbreak. Because normally Ebola virus is a bat virus. Bats or fly fox are the reservoir of the Ebola virus. Normally, the reservoir means that the animal can spread the disease, but don't is affected by the virus. Okay, because the fly fox have a particular immune system that can take a large amount of type of viruses, but can interact with him. It's like a box that transport viruses. In the forest of the Congo, Guinea, or Liberia, normally these animals are eaten. And they hit the fly fox, and also use the blood of these animals like for to feed their body. Normally, the cycle of Ebola infection is bats infect directly humans or beats the monkeys, and after human kill the monkeys and eat monkeys and touch the blood. And also another problem is that for cultural ideologists, when some of the parents or children that die by Ebola, usually they touch the dead body. I don't know why, but they touch the body, and the cycle of infection never stops. And they, during the last outbreaks, actually there is an outbreak ongoing in Uganda. It's not, there are a few cases, but every year, every 2 years, spread this, because there isn't a culture to prevent this type of infection. I don't know why, but WHO go there every year. There are stable labs, but they don't want to change their minds, their way to prevent this. And every time this type of viruses go out. So this is a beautiful chronologist regarding the use of biological weapon like biological agents like a biological weapon. So during the history, so for biological weapon, we can imagine not only virus or bacteria, but also agent that produced by this. For example, Bacillus anthracis produce the anthrax toxin, or fungi, for example, produce a different toxin, or other animals. So toxin of Clostridium botulinum produce the botulinum toxin, etc. In this case, we can consider it biological agent, the toxin. And if you can read, for example, Hannibal against Romans use the venom from a snake to put inside the arrow to kill the people. Also, Romans use Amanita muscaria. Do you know Amanita muscaria, the fungi of Super Mario? Okay. They put the arrow inside the monster of the fungi, and if I beat you with the arrow, I don't kill you directly. If I hurt your epidermis, I put inside you the toxin and hurt you or kill you. But for me, the most infamous use of biological weapons was during 1763, during the British-French war in actually United States, where the British give clothes infected by smallpox to Indians. Do you know that Europeans have the immunity, had immunity to smallpox, but the population of the New World in the North and South America doesn't. And they, like a gift, kill all the trees in the north of the actual Canada. And for example, and there are a lot, can you read it, but the use in the history are fascinating. So this is the explanation of that video. So this is another thing. Do you know, maybe some of you remember the Aum Shinrikyo attacks in the metro in Tokyo? There use this religious cult during that attack use the sarin gas. But some people don't know that first the leader of this cult give some people to Africa to take Ebola samples to use like biological weapon. And another thing, and lucky, that during the sarin attack, also other member of this cult deliberate spread a Bacillus anthracis spores from the different building in Tokyo. Fortunately, during that attack was a sunny day, and the UV radiation inactivated the anthracis spores. if it was a cloudy or a raining day, maybe also Bacillus anthracis outbreak could spread it. I'll say the natural outbreak in the history. So if you say a lot of was a plague or flu virus, one of the, in my opinion, most dangerous was the Spanish flu. Spanish flu was a outbreak of normal influenza and was a zoonosis. And unfortunately, so this is the way that you have to know everything regarding this, is that the few years ago, some scientists discovered the frozen body of Inuit in Alaska died by Spanish flu. They take the sample of Spanish flu, not live viruses, but codify the genetic code of the Spanish flu, because during the epidemics, there's not information, the technology to sequencing the genetic code. The DNA wasn't discovered. And if I know the correct sequence of the entire genetic code, I can reproduce this. And this information in the hand of a terroristic group can create a particular problem. So the next slide, we see I take the information by Spanish flu to give you the information regarding the Spanish flu was a flu, and the flu is we have three different types of the influenza A, B, and C. A and B is the most common influenza that infect human. We classify the influenza regarding the in base of the two spike proteins of the flu viruses, so hemagglutinin and neuraminidase. So we say H1N1 is the type of the spikes protein that are in the envelope of the virus, the variant. I think that normally the people doesn't know. So all over the world, every year, by the influenza diet, normally 500,000 people. Another information. Do you know rotaviruses? Rotaviruses is the good viruses, okay? For us, I stay 3-4 days at home. I drink a lot of water. I take Malox or Imodium and stay. But do you think that in Africa population where the water is not purified and take these viruses every year, diet 400,000 people, especially babies, for dehydration? So we don't consider this, but the orthomyxovirus and rotaviride are a public health problem. And this is the history of the auto. Also, do you know why it's called influenza? It's a Greek story because the ancient Greek, every 10, 20 years spread a particular outbreak of influenza viruses. And sometimes they see when during these outbreaks there was I'll say not many comets, and say, oh, the comets influenza the viruses. Is a funny story. I'll say the name. So also I want to go faster, so especially in this part, but we talk about in the. I don't like follow the slide, but I prefer to make a dialogue with you. So if you want for your exam, you can read all this information. I say that the first outbreak of Ebola starting from 1976, and actually there is another outbreak. The most important, the most famous was the 2014-2016 Ebola outbreak in West Africa that spread very fast and interact with 3 states in the Africa: Guinea, Liberia, and Sierra Leone. Is the most eaten by these viruses. And if you see, if you have a calculator, you see that this is the number of infected people. This is the number of the deaths. More or less the 50%. And there are, so this outbreak is not the most lethal that was discovered. Yes.
Curiosity. With respect to the people that recover from Ebola, do we assist in, like, do we see any long-term effect related to the virus?
The people who were recovered, especially in male, they found first that could be a reservoir of viruses in the sperm, unfortunately, and also can give you a mental health problem. Also, the scars of the viruses, of the diseases. Sorry. So the most recent outbreaks and pandemic all over the world was that SARS-CoV-2. SARS was discovered the first time by an Italian doctor in South Korea. Is acronyms of syndrome acute respiratory. Syncytial acute respiratory syndrome, sorry, and was discovered the first time in China. But the first epidemic starting from South Korea was discovered by Italian doctor. Sorry, I don't remember the name, but died studying these viruses. And it's SARS is part of the Coronaviridae family. It's corona, starting from Latin like crown, because if you see the viruses at an electronic microscope, you see it's like a crown, see to the hub. And another famous virus of this family is the MERS, Middle East Respiratory Syndrome. In this case, so SARS, the first SARS-CoV-2, so the COVID-19 disease, and MERS, all all three cases are zoonosis. The difference between SARS and SARS-CoV-2 is the high mortality rate. So the SARS, the first, will have a high mortality rate around 10%. SARS-CoV-2 more or less 1%. Middle East is too difficult to take the viruses because you have to keep in touch to the reservoir that are camels, but have a high mortality rate around 30%. Please, we are here for this.
About the 19 about the zoonosis.
Yes.
The origin.
Yes. I have my my idea. So, my opinion, this is my opinion. So, we have to take the data. So, just say in the start of the lesson, I talking about that there are flu of dog and cats, for example. Also, there are coronavirus of the dog, cats, bats. Okay. Normally, also me, in the lab of the Faculty of Biology that study your colleagues, I have studied the rotaviruses of monkey and the flu of cat. Normally, in the research lab, we study this type of viruses because it's danger use human viruses. So I can use the similar to create an interaction with human. Okay. In the Wuhan lab, Wuhan, we have to know that starting from there, because Wuhan in the past was forest. If you go there, destroy the forest and build a city in few years. So you have removed flora and fauna and put the humans. There, flora and fauna, it's keep in touch to human. Okay? First thing. Also there was a, no, there is a BLS4 lab that it was built in collaboration with CDC because Americans have most knowledge regarding this type. So the China government take expert to create this lab. But there isn't a culture of this type of research. Okay, have only a lot of money to spend, but don't have the expert to find and to study this type of viruses. Also there, my opinion, normally study different viruses. And for example, study the coronaviruses of the bats or the other animals that stay around in the forest, because building and forest are very close. If you take a not expert scientist that work in a BLS4 lab, and I have to say that work there is a particular work because you have to put the special suites, you have to respect different protocol to not spread viruses. Did you see the movie Lethal Viruses with Morgan Freeman and Kevin Spacey? Do you remember this? It's a beautiful movie that for a little mistake in the BLS4, Kevin Spacey cut his suits and after spread the viruses. So for me, some people say a complotist, the virus was created. Created a virus from zero is very difficult. I can create a chimeric virus. Yeah, after we talk about this, chimeric from the mythologic monster is a part of an animal and other part of an animal and combined. In my opinion, was normally a lab, human lab error, a mistake during a normal research. The scientist was infected. He doesn't know that I making contact with these viruses. These are not human viruses. I go to my family. I go to the shop, and I take a plane to for holidays. The- It's a zoonosis, starting from the bats. Also, scientists have discovered a middle animal that was a pangolino. I don't know in English, pangolino is a mammalian, okay? And pangolino is similar to human. Viruses respect to us can reproduce very fast, can change in error during their replication, jump of species, outbreaks. The problem is that we can defeat nature. It's not, the problem is an ethical problem. Sorry if make philosophy, but the problem is this. You can destroy everything and that you expect that you can change nothing.
Sorry, but this could be occur at any kind of virus.
Sorry?
This can occur, the zoonosis can occur in any kind of virus.
Potentially, yes. Ebola, every virus that I talk today is all zoonosis. All zoonosis. Ebola, flu.
Probably also
Corona.
pox could be.
Could be. Depending on the nature of viruses. And also the other problem are that the transportation in the past, we don't. Today I stay at home. If I take a plane, I go to Australia in 1 day. The problem is this, how we can move. If I move a lot, the virus move with you. If stay at home, I can spread the virus. The problem is this, the virus without us die in few hours, 2 days. So this is how the vaccine of COVID works. But I want to talking about this. For me, this is the most important things. A possible avian flu zoonosis. This is the way how avian flu, so for example, Spanish flu, worked during the Spanish flu. Avian flu is the flu of the birds. Okay? Kill a lot of birds. Okay? But we see in the past and in simulations that if the same virus infect humans, have a mortality rate more or less 90%. I use this number for Ebola, but Ebola, if I don't touch if body fluids, I can take the virus, okay? This is a airborne virus. if I was infected during this lesson, I will infect all of you. The problem is this. Birds have a particular receptor that viruses use to infect birds. Okay? We don't have this receptor because we don't have birds. We don't have, sorry, we are not birds. Okay? Fortunately, pigs have both the human receptors and birds receptor. I talked about this regarding SARS-CoV-2 is the pangolino. Okay? In this case of iron 2 is pigs. Are pigs, sorry. If in China, we know that they have an intensive farm with birds, chicken, and pigs, okay? And live together. Also, humans live close to this. Pigs can infect by birds flu and human flu, okay? Could be that pigs take this type of viruses. The influenza, the name of influenza is the genomic is formed by 8 parts of RNA and capsid. So don't have a unique genome, but it's fragmented. Okay? If I create inside the cell a mix of these, we generate a new virus that can infect and spread human to human with 90% of mortality rates. We are at this stage. If this happens, there are no vaccine, no drugs. There is one drug that can contrast this is oseltamivir. I don't know if you know is the contrast the symptom of influenza. But this is the, in my opinion, the end of the world.
But how can we work in science to prevent such a things? We cannot. Like if it happens.
But the problem also, you have to know first that there are the natural migration of birds. And this is the first. Also, people that okay. Actually, I can block this. The China government, China is not a democratic state, okay? And maybe could be open to WHO scientists. And kill all this, kill birds and pigs, and burn the bodies.
Yeah.
The only things. Fire destroy the viruses. Fire or UV is the two things. So this is the other not mortal outbreak that occur in nowadays. Do you know the problem of the norovirus in UK hospital? Norovirus is like rotavirus, is gut viruses. But in UK, that find in the end of 2024 a new strong variant of Kawasaki norovirus that are particularly aggressive to immune system. Also, the human metapneumovirus that's discovered in the in the start of 2025, starting from the China and discovered also in Italy, and it's called the Jubileum viruses because we discovered during the start of Jubileum from the tourists that starting from China to Italy. Is another virus that can spread and create a pandemic. So this is the other problem regarding the if I know the genomic sequences of a different type of viruses, I can create the chimeric viruses, for example, and I leave you for the long break. If, for example, this is only the fantasy. We know that Ebola is not airborne viruses, okay? They spread only with the body fluids. Good, fortunate, but have a high mortality rate. For example, if I create a virus with airborne like cold, the normal cold, il raffreddore, okay? And put inside the genome of Ebola, I have created the perfect weapon. I can kill millions of people in 1-2 weeks. This is the problem of the. If I have a bioterroristic agency with that know and have genetic engineering scientists, we can create everything and use to create a lot of panic. So this is the end of the first part. Maybe I bored you. Sorry for my pronunciation. And the second part, so easy. We talk only the fungi, but the most important thing regarding the possible way to create a biological weapon, in my opinion, are the way of spread of viruses. See you 10-15 minutes, okay?
They would know it's September.
Devo dire, io ho fatto lo sprint.
Ciao, Franco, buongiorno. Allora, questo è un sì, grazie, mi trovo
Sì, un attimo, però io ho fatto i miei a pranzo, però certo, certo. No, vado in corridoio. Va bene, cavolo,
Yes.
Visto che la generazione non viene, allora rientrava.
No. Questo sta pensando.
Ma questa non c'è vita.
Yes. Yeah.
So. That application is pretty cool. Fine. You can say all of the issues about you had on the best. And my last thing is everything was covered. I'm joking. They walk the women. Women get, they get really fast. Really, you're like, at least, the back impression. You want to lie, it's wrong. It's wrong. Easy go, not really very much. People want lots of occasions. That's going to happen. Not a good day. So one other thing. The one is big. No, I'm just looking at I'm getting it still on. So, if you're interested, I can give you a question for the answer. I talk to you. I can't give it.
Okay.
Then, why? That's fine. Because so, family or what, they were the biggest, that's wrong, that is back to. They want more, they have goal, the 25% is all about 5 hours, you only have one. But also, they need more with. Well, I need that base, but do it. If you want to, the big one, a real one. You know the test. No. It's not a all there. It is rather, I have to buy house. Which time? You live in grants, always. Sure. Okay. Hello. It's not Application is signed off for 2 weeks. Tanto I don't already have my certificate, but I are already devo che la si contraria. Only if you come out to let us know over. We will. Cynthia, right.
C'era una compagna di scuola che da riprende quando diceva nata totalmente incendio che non fanno vedere. E non è fantasioso io un po' un gamma, perché quando gli schiavi che demanda è un iscritto che fai si chiama il laudo però è bello è mai sicuro delle cartelle delle 20 pagine le lauree della vita che non devo guardare che c'hanno la questa c'hanno Paolo molto forte. Cosa importante. Che si trasmettono in maniera quello non è scuola di questo prima. Quelli che sono a produzione diciamo ci sono periodo di solid che si parlava più
Allora, perché quello cioè, l'interno della gestione per cui il federalismo è più buono, magari piano poi altre. No, it's not, it's not that it's so, well, okay, in perspective, it's not being all the time. You know, I said, I tried to French a little bit better, but it depends on how, you know, the level is required. Because if again, say, you speak with no, and so that you get a small level of people to speak, something like that, people are speaking. No, I see that. Altamente sono Ah, okay. Maybe yes.
Sorry, regarding viruses. Regarding viruses. Do you have question? Sorry, silent please. I don't ask you if you have a question because during the lesson I stop and I prefer this because it's the most interactive lecture. But in the end, if you have question, please, I am open to you.
If I'm not wrong, there are viruses that can infect other viruses.
How?
The professor told us in the lecture.
Viruses?
Viruses that can infect other viruses.
Which virus? I don't know, maybe during the first part of lesson, I talking about a gigant virus that is in the middle between a normal virus and bacteria. But if you think how, if a virus is infect another virus, how is the way to replicate itself? The virus don't have the structure to replicate itself. So, for example, you are a biologist. We can talk the same language. If I have to replicate it itself, it needs amino acid. It needs azotate bases. It needs lipids. No, lipids, no, protein. Maybe the structural basis, sugar. Okay. Maybe there is a bacteriophagist that is a varieties of bacteria. Okay. The bacteriophagist, T4, T3 is the way that we study the first viruses. But I don't know, maybe we'll discover a mechanism. The research is in continuous development. But I didn't hear about this, but maybe I'm wrong. I will search in literature and maybe I will find this. The new concern regarding the viruses and the social problems are this chimeric virus and also the zoonosis problem, because we see COVID or SARS-CoV-2. During the coffee break, talking about that now I take the COVID and I have a strong cold, some fever, because this virus is after generations, it's endemic in human population. Now it's endemic.
Now we can talk about zoonosis because now there is a human SARS-CoV-2. But the first variant was a bat virus. The problem is when during this so after the coffee break with your colleague talking about that in USA there are a lack of eggs. Do you know why? And they buy more tons of eggs from Veneto region in Italy. Did you hear about this? Because the US government had to kill a lot of crops of chicken because they found avian flu outbreaks. And if avian spread, also infect the birds and kill the fauna, also could be infect the human and create a human outbreak. And fortunately, the mortality rate of avian flu is around 90%. The only 10% is because are lucky and have immune system, because the nature create a immune system to preserve a part of the population. But the other 90%. So, for example, another it's beautiful to talk this because of my passion. Did you see the film, the movie I Am Legend? Do you remember how start the outbreaks? With virus. In the start of the 20th century, Italian doctor find a way to destroy a type of cancer using herpes viruses. I follow the virology research because when I was a student in USA, for example, I want to talk about this because I want to give you the pills and not make a normal lecture. I give you the standard information because for you this information is much in deep, but you don't need this. In USA, in the start of the 21st century, discovered during production of the bovine broth that we use in the research had infected broths. Okay, this broth is used normally in virology research and for grow the cancer cells. And a lot of this was infected by a viruses, and the factory that was product don't recognize this. So during the normal research, the scientists see that the all cells died. Why? Investigation found not human viruses that destroy the human cancer cells, because normally the human cancer cells produce different receptors. Okay, the locket and these viruses attach only the cancer cells and destroy the cancer and don't infect the normal cells. If a pharmaceutical company find this, have find the cancer cure. But the problem is, are you sure that you keep the control to these viruses? Are you sure that these viruses can spread to other people, rearrangement, and create a new disease? Is a problem. The new solution that, for example, Moderna, regarding the study of vaccine, is created a personal vaccine to cancer cells. So I have a cancer and Malizia have the same cancer, for example, but we have two different persons. And the future, in my opinion, is to create a personal therapy to destroy this type of the cancer. For every cancer that's solid cancer, that the progress of medicine is evolving every day. So now so this is a presentation that I have done with Nicola Simone. Do you know Professor Nicola Simone? Maybe give you different.
Monday, yeah.
I have done this presentation with him, and Nicola, my friend, have a particular way to create a joke presentation. So just say before, I don't want to bore you, and I know that you have all of the other 4 hours of lecture in the afternoon, so I won't stay so quiet during this second part, and I won't make this presentation like a chat with you. So just say before, another biological aid, all biological weapon starting from biological agents. So just say before, I say that not the microorganism have to consider it biological weapons, but their production could be. And for example, the toxin from plants, bacteria, and also viruses can use as biological weapons. Now we talk about the fungi and the toxin from fungi, but normally also we have the toxin from plants. Do you know some plant toxin that can use like biological weapon? One is most famous, ricin, also abrin, or other. For example, rotaviruses, this is a pill, rotaviruses, and is the way that you have a gut problem, is the only virus that can produce a toxin. And the way of this, also the infection, the toxin is the non-structural protein 5 of rotavirus, is a toxin that can, I'll say in English, bucare, perforare the gut cells and create the problem regarding of disease. During the replication, the viruses create itself a lot of virions, and these proteins, viruses don't use it, don't take this protein in the virion, leave the protein in the cells and make porous and destroy the cells. Physically destroyed. Also, the cells is destroyed by the budding of the cells. So a two-way to create a problem and the consequence of disease. So the agenda of the fungi is a fungi, mushroom, and all fungi. So the real agenda is fungi and toxin. There are fungi, you know, in the biological world is another kingdom, because we have plants, bacteria, animals, and fungi. Fungi is a kingdom itself. So there are more than 3 million of fungi species all around the world. 3 millions. And also the biggest organism in the world is a fungi in the United States. It may be, I don't know how it much, maybe a lot of kilometers square. It's one fungi kilometer square, a lot of kilometers. Is maybe in the Yellowstone Park, maybe, yes. So fungi have a different way to show us. So we have like a single cells or a biggest, like in the picture, but is the same family. And depend on the species, you can show in this way. Normally are eukaryotics. Do you know what I mean when I say eukaryotics cells? Can be monocellular or pluricellular. A famous monocellular fungi. Do you know?
Yeah.
Saccharomyces cerevisiae, or another for a disease that is not good for human, Candida albicans. Do you know? La Candida is a monocellular fungi, or pluricellular, the fungi that we eat normally with pasta, okay? And the way of reproduction can be asexually or sexually. So, like a bacteria can reproduce itself from a cellular, or maybe can interact with a female and male of the same species, DNA recombination and create a new fungi, like humans or animal in general. Fungi, just say, can be single-celled or very complex multicellular organisms. They are found in just about any habitat and can live in the land, in soil, or in plants. And something of fungi also have discovered in the sea. The fungi are essential to the recycle of the organic material in the world. If we don't have the fungi, we don't have the normal life in nature, because a type of fungi are the only that can digest the diet tree, so digest the cellulosa and create a recycle of carbon in the nature. Some are parasites of plants. So, for example, I don't know if you like wine, but in the start of 20th century, a fungi could destroy all the wine tree in the world. And so this is the way, because the wine tree in all over the world are divided in two parts. We have the tree, but the roots are of different species that are not immune to a particular fungi. Also, no, quello è la peronospora is a fungi that can attack the plants, and you see on the leaves. The leaves became yellow. Okay, brown, yellow, right? But it's called in Italy piede franco, because if you take the tree, the Barbatelli tree is it's divided in two parts. The roots from America and the stem, the stem is from Sangiovese, Chianti, Pinot, blah blah blah. Okay. A very small number of fungi cause disease in animals. In human, that include skin disease. So in Italy, we say, I have taken the fungi. It's called the athlete's foot, or normally when you go to pool and take this fungi that is the same family, or trash. So I don't want to bore you regarding the life cycle or fungi. It's simple to show this picture. So normally we have this, the spores. The spores make germination, so grow up in few words. In this case, we have two ways. When grow up, create a mycelium in mycelium. Mycelium from living. If take a sexual union with other mycelium, so we have mycelium male and mycelium female, create a sexual fruiting that we see in nature, and we eat the sexual fruiting. Or the other way, the mycelium create a sexual fruiting that are haploids and not diploids, create the conidia that are asexual, conidia germination, and make the cycle from starting. This is the way. Asexual or sexual. This is important. You have to know. The fungi, in general, it's divided, for example, in molds, yeast, lichens. Lichens are a beautiful thing in nature, that there is a collaboration. So when we're talking about parasites, the normal definition is that the fungi take the nutrition from the plants and stay all, so it's like an infection. Take all the nutrients and don't do this, don't do nothing. Lichens is a cooperation. So fungi take the nutrients from the alga, that take the nutrition from the sun and create the sugar by photosynthesis. And in the same time, the fungi from the soil take mineral and other nutrition to the alga, and this creates a cooperation. Molds, maybe you, in the bath, when there is a humidity or when the fluid is along, we don't eat. The molds is particular because for the human health is a problem, because for example, there is Aspergillus, is the molds of the fruits, for example, that create the spores. These spores can go inside our lung and create lung problems, especially in people who have not a healthy immune system, or problem of immune system, or lung cancer or other disease, this could generate an incrementation of the respiratory problems. But in a health people, the immune system attack directly the spores and destroy it. Yeast, so we talk, most important for the production of the beer, for example, or pizza, is the Saccharomyces cerevisiae. Candida albicans, fortunately, can create a disease in the oral or sexual reproduction. And normally are not pathogens, but in particular condition could be. Lichens, just said before. Macrofungi are the fungi that we see, and our imagination when you think about fungi is the macrofungi that we can eat. But as you can see in this imaging, there are fungi that can produce bioluminescence proteins, and there are a lot of species. And this fungi that talking about is Amanita muscaria. Is the, do you know it's called muscaria, the name? I love the name of thing because the name explain. I remember that muscaria from mosca, so fly in English. So use it to kill, it's like a repellent. Was used for this. It's called Amanita muscaria. Is a very toxin, and if you eat a small pieces of this fungi, your liver is destroyed in a few hours. So this is rough and smooth. So is a heart disease, or especially in plants, and in this case, it's the nutrition from the corn, and is a parasite of plants. Mycorrhizae, the most famous is truffle, right? The mycorrhizae is a way of the cooperation between fungi and plants. Because just a similar to lichens, in this case, the truffle take the nutriment from the roots of plants, and in the same way, truffle gives specific mineral to roots to grow up the plants healthy. Toxins. Toxins, the definition is that are substances produced by living organisms. Okay? The definition is important. Generally are protein or peptides, so a little protein, peptides. Generally are developed usually to protect the organism from pathogens or predators. So this is important to know how many toxin, toxicant, or poison. In Italian, especially in Italian, we usually confuse these terms, or is the same, but it's not this. Because toxin is a specific substance produced by living organisms and the effects are immediate. Toxicant, for example, are chemical compounds that can injure humans. The duration and the effects is different in the time. For example, if I am in contact with a lot of type of chemical compounds and the effects I will see after days or weeks. Okay, if I'm making contact with a botulinum toxin, I will die in few hours. Chemical compounds, not. Poison is important. A toxin or toxicant that can cause immediate adverse, and a small amount, like a poison of the snake, for example, is usually to cause problems to human health. Sorry, can you hide the voice? I have the... So for venom, in Italian it's the same things. Velenoso, okay? But for venomous, if I want to give you this slide because it's more simple to you, but in few words, if something bites you and you die, it's venomous. If you bite something and you die, it's poisoning. Okay? Venomous snake, poisonous fungi, for example. Okay? So a snake could be venomous. A plant, if have specific toxin, is poisoning. Some fish can be poisonous and venomous. And this is famous, the pufferfish. Do you know the story of pufferfish in sushi and Japan cuisine? This beautiful fish, very beautiful. In Japan, it's cultural because it's a very particular tasty for the Japanese sushi cuisine. But a little bit part of this fish is not poisonous. The other part of fish is poisonous. And there are a school of master of cut fish that you need years and years of experience to cut in the specific this fish, because inside the poisonous part of fish is produced the TTX, tetradotoxin, is one of most poisoning compound in nature. Okay?
So these should be considered as a weapon.
Sorry?
A biological weapon with tetradotoxin.
Always. It's used by Soviet Union during the Cold War.
Ah.
Also, we live in the West, so we don't know if they always is the truth. But normally, one thing, the report of the CIA was that during the Gulf War, the first Gulf War, Saddam Hussein have a lot of tetrodotoxin or aflatoxin stock pills to use like a biological weapon. After we talk about this, it's interesting. Another thing that I want to say to you, maybe other professor say to you, but when I can say that one thing is toxic. If you think everything are toxic, I make you a beautiful experiment. Water. The amount. The amount the toxic depending on amount. If I drink 5 liters of water in 30 minutes, I will die. But water is the base of life, not toxic. Predict our blood and give you an aneurysm. So now there is a list of the organisms that produce toxin. Every organism in the world produce toxin. From bacteria to mammalians. And I show you some examples. Also the birds. In Papua Nova Guinea, there is a bird that inside his neck produces a toxin that he defends from the natural predators. Bacterial toxin. The most famous was botulinum toxin or trachis toxin. Viral. The only there is a mistake in this slide that make Nicola. But the only viral protein in nature is the non-structural 5 protein of the rotavirus. This is an important slide, is the classification of the bacterial toxin. Bacterial toxin can be divided in exo or exotoxin or endotoxin. And this is the different properties. The most important in the drugs production in a pharmaceutical company, so I can tell you that is the working this way, and protect the people from this endotoxin. The endotoxin, let it reach, is stable at 250 degrees. So if you don't kill the bacteria, the protein that produce the bacteria kill you. And the only way to produce drugs without this, every drug is a 300 degrees sterilization of vials that can destroy this, or using chemical compounds, but the problem is remove the chemical compound. So in this case, we use hot water that can destroy. Phytotoxin or plant toxin. The vegetable kingdom is by far the largest reservoir of toxin. So the most toxin, the large amount of toxin produced in the world is plants. From algae to tree, the number of plants capable to produce one or more toxin is simply large, and it makes no sense try to analyze them. There are very large amounts. The most important and used also in medicine are curare. You know, curaro is used in the past to to block the
Coagulation.
yes, resin, conin, aconitin, abrin. There are a lot. Other toxins that are present in nature and are very important for the health problems are the mycotoxin. Mycotoxin, just suggest the name, is the toxin produced by fungi. This slide is a representation of the most important mycotoxin in the nature, and the most important that maybe you know the name is aflatoxin. The problem is aflatoxin. The problem that, for example, in China, have a biggest problem or biggest liver problem because they use a large amount of rice and are not conserved in the right way. Not conserved in the right way are the humidity. The humidity produce fungi. The fungi produce aflatoxin. And every year, people eat a large amount of rice contaminated by aflatoxin. Aflatoxin destroy liver cell. Another aflatoxin that make problem, especially in the west side, is the milk aflatoxin. Because for the globalization, the cows eat the crops from these regions. The people, the cow eat contaminated foods, and when we don't have a metabolic way to destroy toxin. The only is create a vacuum that can expel. But normally, when I eat, I remove it in the same way. If I eat pasta, for example, my metabolism convert to glycol and use for energy. Toxic? No, we don't have a way to destroy the molecular compounds. And in this case of the milk aflatoxin, the food contaminated is metabolized by the animal and go to the milk. I drink the milk with aflatoxin. It's a recipe. This is a slide that for chemical maybe is interesting, is the representation of molecular structure of different aflatoxin. This is the peptide structure of a mycotoxin, in this case mucoricin. If you want go in deep of this argument, I suggest you to read this beautiful review regarding the whole list of the all mycotoxin in nature and their function. This is in this slide, we see the modulation of the host immunity by mycotoxin. So normally there are two ways. The first, that the toxin destroys directly the surface of cells and create a hole and the cell destroyed, or block the replication of DNA. The cells can reproduce itself, die, and consequently die, and causes the necrosis of tissue. Another way is that mycotoxin use destroy the interaction cell by cells. You know that in a tissue, all cells is linked. If I destroy this bridge, the cells are free and the tissue go to necrosis and die. And the die of entire organism. So now we are in the heart of this lesson. I won't leave you more times to question and to restore your brain, because maybe we are in a Wednesday in the middle of week, maybe you are so tired. Yes.
The snake bite the mice, the rat, what we, after we eat the brain, and the toxin that produces in itself are not metabolized by the snake, or the snake have a particular metabolism.
Generally is a compound that produced by snake is not the effect for it, for the snake.
Yeah, by
Yes, I know what do you mean. Yes, no, in
per capirci, c'è ancora la tossina.
I remember regarding, because the venom of the snakes works in two ways. One create gelification of the blood of the la preda, the mouse in this case, and the snake eat after a few times. But I don't know if it's an important question. I don't know if it's metabolized by the snake, but maybe yes, because if you think is venom, if don't use it, is replace it every time, no? Maybe we will. I will inform about this. Yeah. So I can tell you that in the human history, mycotoxin are used like biological weapons. Fortunately, produce a large amount of toxin to create a big attack is very difficult, because we need a biostock and purify the protein and have a large quantity that you can use. You can create this inside not a regular lab. You have to create a specific factory to create this, and it's very difficult, fortunately. But as we will see, if I want to kill, for example, one people or few people of person, I can create a small amount of this toxin in house, for example. The most interesting toxin currently considered as biological weapon agent are, you have to know this, botulinum, ricin, saxitoxin, and abrin that are not listed. Ricin and saxitoxin are currently enlisted in Schedule 1 of the Chemical Weapon Convention, are listed there. The term of mycotoxin was coined in 1972 because is an example that I have done before regarding the food contaminated, because in England a large amount of turkey died by eat some foods. It's called disease X. unknown disease of Turkey. After investigation, they found that the foods of Turkey was contaminated by toxin, in the specific by Aspergillus flavus, that are a fungi. So this is the most considered attack to using biological, mycotoxin like a bioweapon is was created like yellow rain. After the Vietnam War, the Soviet army used G2 toxin combined with the clouds, and the toxin was dispersed in the rain and infected the army hidden inside the forest of Vietnam to kill people. Crazy, yeah? But they use this. I leave you with this. All mushrooms are edible, some are only ones. This is a list of publications regarding this, biosafety, biosecurity, and toxin, and the use of as a biological weapon. Another thing that I didn't see. We say that the fungi produce toxin and something that can hurt us. Okay. But in the past, thanks to mushrooms, we found a compound that saved millions and millions of people. Penicillin. From penicillium. You know the history of discovery of penicillium? It's famous by Dr. Sir Alan Fleming. I don't want to bore you again. If there are questions or curiosity regarding. Question is only one, only for us.
The methodology is good. The methodology is responded by the science.
Yes.
The question for this. We use that as biological weapon to affect
The problem is the time. A prion it's not my field of study, yeah, please. But I don't know how many times the prion is active to make this. For example, one biological attack documented, in my opinion, was a stupid attack. But in this case, you see the lack of the control. Is using salmonella in during 18 in USA. I don't know in English, setta, okay? To don't make the election of a major in a city in USA. Inside the restaurant, put the salmonella bacteria inside the salad. People go there, eat, take salmonellosis, and go to and remain at home because it's impossibility to go to election. In these cases, I can say, okay, it's not an effective attack, but the bacterium of salmonella can resist a lot of days in good condition. Prion, I don't know, because prion is a protein, and maybe with the heat or the condition of storage can determine the quantity. But the use of prion like biological weapon, we don't have the documented, because it was a recent discovery. But we will see. We can see if it is a stable compounds, maybe could be used like biological weapon. In my opinion, in general, the mass destruction weapon, the most effective is virus. Why? If I want to destroy my enemy, another state. If I use a nuclear bomb, I destroy everything. There is fallout, etc., etc. If I create a virus, and also I have the cure, I have the vaccine, and infect you, I kill you all the people. I will not infect it because I have the drug or vaccine, but all buildings, all structure, all the resources remain there. I can take it. Chemical compounds, more or less, because you have to use something that can removed and don't stay and don't make an effect in the future. The problems, for example, Ceres, no? Do you remember Ceres? The dioxin, okay, it's a chemical compound, can create problems, but you can remove it. Stay there. You have to take it and put to the other part, but you can remove it. Viruses, when make its effect, die with the host. Another questions?
Just a consideration.
Please.
But seeing a moving Mission Impossible, I don't remember the name. 2 o'clock.
Bellerofonte Chimera. As the example that they have done.
Technical point of view. If I made research to develop a virus or a genetic modified virus, it was strange for me seeing this kind of movie that okay, I have the last virus in the vial, and if I destroy it, but in my opinion, there was in somewhere protocol. For example, if I'm developing a virus, I leave the protocol to how produce it. No?
Yes.
So for me, it's strange that often they talk about, okay, this is the last vial and the last. Because in my opinion, the creation of this kind of weak, in this case, is not instantaneous action or stochastic action that I'm very lucky, I find that there are some knowledge process, and after, okay, I use the vial, but I know how to reach.
The movie.
Yeah, obviously.
No, it's the movie, okay, but it's smart. Huh? The problem in the movie is not the problem. the storyline is very smart because in this case the virus was development to create a problem. Okay, I have the cure, and I will make lot of money.
Yeah.
The maybe I remember they have a small amount of the virus.
Yeah.
Okay.
Yeah.
But it's normal for two ways. The first, to create other viruses, I have the host. So if I spread the
Yeah.
you are the bioreactor
Yeah.
to create a large amount of viruses first.
Yeah.
But the smart is that I need a biggest factory to have a lot of quantity of viruses, but I don't need it.
Yeah.
I need a little amount.
No, but
But in the movie also, maybe I remember one have like a gun to put the directly viruses inside. The other that Tom Cruise destroyed, the first, was the flask to replication, because a little amount of replication, the virus need, because after the virus died. So they have a little lab to take just the quantity necessary to create it. But I don't need a large amount because do you remember maybe the female protagonist? If only one person inside of the city can create a pandemic in few days. But another stupid thing is that the viruses kill you in few days. This is another thing because, for example, I say, in your opinion, which is the virus most smart?
It kill them, but in long term, so that you have the time to spread it.
yeah.
Ebola is a stupid virus. Kill you in few days. The most, in my opinion, smart virus is herpes simplex 1. Because, okay,
Yeah,
Herpes.
for the people in the world.
90-80% of human population are infected by virus that you have in your ganglion and stay there in relax.
Very efficient way to survive.
And spread out when your immune system is have a luck.
Yeah. No, but my question was different. And I believe regards us because we are also thinking to prevent this kind of event. No? If I am a terrorist, okay, and I have a lab to produce a small amount of virus, I believe that I store the research result. Okay, to produce the virus, I have to follow this step. Okay. If someone destroy my final virus, but I know the procedure to produce the virus, I don't need to new virus. I know the procedure to.
You have the recipe.
One,
You
yeah.
have the recipe. Yes.
Yeah, it's strange that in this kind of things, no one take the aspect of the research process. If I know how to do, and I know I have to start from this first virus, I have to modify with this process, I have to, and so for our point of view, is the worst situation is that people can
Take the information
to this kind of information because the development was done by highly experts because they had to find the way. But if I have the procedure, I'm common people, I can reproduce this.
Maybe in the movie they assume that is a natural outbreak and the only one, and you don't have information to replicate. Create ex novo viruses, maybe it's very difficult.
Yeah,
Ex novo.
yeah, it's a modification.
But in the future, maybe. I said before regarding the avian flu that found in the Inuit in the permafrost. There is a publication from the Bacteriology Netherlands that give to, if you go to PubMed, you find the genomic sequencing of the avian flu. It's a study from the 2013, 2014, maybe 10 years ago. Is a lot of information that bioterrorists that can use for scopes. We will see. Maybe the actual problem was the natural outbreaks. Because the intensive crops, the intensive and the live very close to the animals. The history repeats. In China, there is country. There is country. In China, a normal farm, not a big, normal farm with the farmer. There is a first floor. I see it. There is a first floor. Pigs and chicken live together, and they live after. Like in Italy in the past, when created the La Stalla with cows in the first level to the pit to restore. But there is the transportation of the human movements that stay 100 years ago. Today, stay here. In few hours, I stay in the other part of the world. And the contamination is strange. Another, only to talk, another beautiful use of biological weapon in the literature is, do you know the writer Dan Brown that write Da Vinci Code? He write another book, it's called Inferno, Hell in English. And then the problem was the large amount of the human population. We are too much in this world, and we don't have to reserve the resource to live food for poor people and rich people. In this book, there is a scientist that create a new plague. I don't want to spoiler you, but this new plague really is a virus that don't kill the people, but make the half population sterile. And the beautiful is that he leaves some way to found these viruses, and it's strange because when found the source of these viruses is broken from 2 weeks. And in 2 weeks, all human population are infected. It's not a strange thing. Could be. Could be. If you live, we are in Italy, no? Okay. Try to spread a disease in the center of Rome. how many tourists, how many people from all over the world. In few, not in few weeks, in few days, the disease spread. The problem is this. If you can containment and create hot zone, warm zone, cold zone, okay. But in this case, it's not under control. You can try, it's not under control. The only thing is if you have to launch and create a drugs or a vaccine that can save most people, but the probability that the people die is very, very high. So, thank you so much. I hope that don't bore you too much. Sorry, I repeat for my pronunciation.